Namaskaram. In this session, we are talking about the structural features of normal secondary growth in dicot stem. So here we can see the structural diagrammatic representation of the dichotomous stem showing secondary normal secondary growth in thickness. Here we can see the cross-sectional view, and it contains all the tissues which are getting differentiated. And in primary structure, we study about only the broken ring of vascular bundles which is present, and it will show the formation, presence of a white bit in primary structure, and there you can see a limited number of broken ring of vascular bundles, and it contains only fascicular cambium. That is, the cambium or the vascular cambium is only present in between the xylem inside the vascular bundle, and it is called broken ring of vascular bundles. And here in primary structure, we can see it contains a wide cortex, and cortex is usually heterogeneous in nature. That is, it contains either parenchyma, collenchyma, chlorenchyma, and also parenchyma cells as in primary structure. And ultimately, the outer epidermis with the thick cuticle is present in a primary structure of plant body. But coming to the secondary structure in dicot stem with a normal secondary thickening, we can see the presence of all these tissues except some of the tissues which are getting disintegrated, as the cortex it contains some modifications or some changes we can see in the cross-sectional view, and similarly in the vascular region there also we can see some modifications or changes, and it contains entire or reduced the pit is there at the center, and similarly the inner portion or the major portion of the stem in the cross section we can see it contains the xylem, one of the complex vascular tissue, and it occupies the major portion. And similarly here in secondary thickening we can see there is a continuous ring of vascular cambium which is developed by the union of fascicular and interfascicular cambium develops to form the vascular ring of cambium. And there is outer to the cambium it contains a complete ring of the phloem elements, the another vascular tissues in the plant body, and the cortex which is getting reduced, and somewhat it contains the secondary cortex, and sometimes it contains or some of the examples which contains only the chlorenchymatous cells. The remaining sclerenchymatous cells or even collenchymatous cells or parenchyma is getting disintegrated due to the extrastellar thickening in the plant body. And similarly, the outer or peripheral region which show the differentiation into the so-called formation of periodome and lenticels, which develops the another or new group of tissues which is called phellem, phellogen, and phellodome, and the formation of the core tissue in the outer region. So, coming to the structural details, from outer to the inner region, what are the tissues or the components which is occupied in a secondary growth in a dicot stem? So here we can see the outer lenticel. It is peripheral outermost layer of the stem showing the broken areas here and there in the stem, and it is called lenticels. It contains loosely packed thin-walled parenchyma cells. And the another component in the structure, it is called periodome, and actually the periodome is the outermost layer, and it is a part of extrastellar secondary thickening in dicot stem, and the periodome contains the outermost dead layer of cells which is called phellem or cork, and inner to the phellem the middle Phylogen, actually it is the cork cambium. It is actively dividing meristematic cells which derive outer phloem or cork and inner phloem or secondary cortex. So inner to the phylogen or cork cambium, you can see involved loosely arranged or the cells with the intercellular spaces, somewhat round or oval shaped phloem or secondary cortex is there, which is developed during secondary thickening. And innermost layers sometimes show a number of primary cortex with the parenchyma cells, and in the inner portions of or in the middle portions of the cortex, what we can see here and there are patches of sclerenchymatous bands are there. It is called bundle cap or sclerenchyma. It is nothing but at the early stages or in the primary structure, we can see the broken ring of vascular bundles which are capped with the sclerenchymatous bundle sheath, which is called bundle cap. And actually, due to the secondary growth in thickness, the primary bundles which contain the sclerenchymatous bundle cap, which is getting pushed outer to the cortical region. And so in secondary growth, we can see there are patches of the sclerenchymatous bundle cap. And inner to the bundle cap or inner or the next layer is the continuous layer of phloem, and it contains outer portions with the primary phloem, and the inner portions are occupying the secondary phloem, which is derived from the vascular cambium. And here in the secondary phloem, we can see the different elements of phloem components, and they are called the phloem fibers. Some of the cells become dead and thick-walled, which helps to protect the congestion or disintegration of the phloem cells due to the secondary growth in thickness. And phloem fibers are the components in the phloem. And here you can see the secondary phloem, and also the secondary phloem consists of sieve tubes, then companion cells, and sieve plate and phloem transverse cells. All the derivatives of phloem is formed during secondary growth in thickness. And here you can see the phloem and xylem, that is the most of the portion is covered or developed with or occupied with the xylem elements during secondary growth, and xylem and phloem is getting demarcated with the continuous ring of actively dividing cells, which is called the vascular cambium. And the vascular cambium is nucleated, and they are living cells. They are actively dividing cells inside the secondary growth or inside the stem. And from the vascular cambium, coming to the phloem, what we can see from the cortical region, that is throughout the phloem and through the vascular cambial ring and through the secondary xylem, there are some thin-walled unilayered parenchyma cells which towers from the phloem, that is in the base, from the base of the secondary cortex up to the pith region, which is called the medullary rays. That is, medulla means the central portion. Ray means it is an elongated layer of cells passing across the medullated region or medullary region, that is the center region, that is from secondary cortex to up to the pith region, and they are called the medullary rays. And coming to the next characteristic features, that is the central portion, the medulla, which occupies the most of the area, is with the secondary xylem elements. And in secondary xylem elements, we can see it contains the vessels, that is xylem vessels, and also the secondary xylem contains tracheids. We call the dead layer of cells, which is called tracheids, and also the secondary xylem contains the xylem parenchyma, and also it consists of the xylem transverse cells, and also it forms the xylem fibers. So all the derivatives which are derived during secondary growth in the one of the complex vascular element which is called the xylem. It contains the cells, tracheids, xylem parenchyma, xylem transverse and also the xylem tracheids. Okay, and here we can see the intraciliary conjunctive parenchyma. Conjunctive parenchyma means whether they can differentiate into actively dividing cells or otherwise they are living cells which is present in between the vascular regions. It is that is the xylem is housed with such thin-walled parenchyma, elongated parenchyma cells which are intercalated with the xylem elements, which is called the interciliary conjunctive parenchyma. And coming to the differentiation of the xylem during secondary growth, we can see the wider vessels, that is the metaxylem vessels, which is present in the secondary areas of xylem, and it also contains some protoxylem elements and which are usually placed inside the innermost portion of the xylem, and they are called protoxylem. And metaxylem are derived during secondary growth in thickness, and protoxylem are derived during primary growth in dicot stem. And this primary xylem, just you can see, it is almost in the enda nature. That is, the protoxylem towards the pit and metaxylem towards the periphery. And it indicates the stem character, and which is actually developed during in the primary growth in thickness, and due to the secondary growth in thickness, more number of metaxylem vessels which are formed and the xylem elements are differentiated. The primary xylem is getting pushed towards the interior region, and usually at the center we can see the presence of thin-walled parenchyma cells which are called the pit. That is, the center portion contains a limited number of oval or rounded thin-walled living parenchyma cells, and at time what happened, the more and more secondary growth takes place, sometimes the pit is getting disintegrated and we can see as a hollow region, or otherwise it is totally absent because the xylem which is getting occupied at the center region. And this is the normal secondary growth in dicot stem, which we can see through a cross-sectional view, and here I am representing here the diagrammatic structure of normal secondary growth in dicot stem. Okay, thank you.